Across the animal kingdom, broadcasting distress is a matter of survival, and it is almost always loud. A separated pup whimpers, a startled bird shrieks, and a lonely wolf howls to signal its location. Humans stand entirely alone in their physiological response to pain. We are the only species definitively known to shed visible, saline fluid from the eyes in direct response to our internal psychological states. This phenomenon baffled early scientific inquiry. Charles Darwin, despite his revolutionary insights into human emotion, famously dismissed emotional tears as a purposeless byproduct of facial muscles contracting during moments of distress. From the perspective of 19th century natural selection, weeping looked like a counterproductive glitch. It expends vital hydration and temporarily blinds the organism, visibly broadcasting vulnerability to any nearby predator or competitor. Modern evolutionary biology and neuroscience have since dismantled Darwin's conclusion. Contemporary research reveals that the human tear is a targeted survival mechanism, orchestrated by the brain to manage states of overwhelming emotional arousal. The process begins deep within the brain's emotional engine, the limbic system. When an individual experiences profound grief, sudden fright, or intense social rejection, the amygdala processes the emotional significance of the event and flags it as a crisis. The amygdala transmits these signals to the hypothalamus, which acts as a physiological bridge. From here, regulatory signals descend through the central autonomic network, targeting the brainstem to trigger an emergency autonomic response. Functional neuroimaging reveals that the brain makes little distinction between physical injury and social anguish. Both experiences recruit the same neural circuitry in the pain matrix, including the anterior insula and the dorsal anterior cingulate cortex. Because early humans relied entirely on social cohesion to stay alive, a broken bond was treated by the brain with the same neurological urgency as a broken bone. It is an immediate threat to physical survival. This anatomical overview shows the human lacrimal apparatus. When the command to cry reaches the brainstem, signals are sent directly to the main lacrimal gland. This system is separate from basal tears, which provide continuous lubrication, and reflex tears, which wash away smoke. While those maintenance systems respond to external triggers on the eye's surface, emotional tears are generated entirely from within. They operate on a divergent neural highway, driven exclusively by internal affective states. Because they are triggered by the brain's stress centers, emotional tears possess a distinct biochemical signature. They are highly concentrated with stress hormones like ACTH and leucine and enkephalin, an endogenous opioid that acts as a natural painkiller. Metabolomic analysis shows the chemical profile even shifts with the specific emotion. Tears of joy exhibit higher concentrations of dopamine and oxytocin, following different metabolic pathways than tears shed in sorrow. Each emotional tear carries a deliberate biochemical payload, specialized to the very specific internal state of the individual. In the predator-dense environment of the Pleistocene, acoustic distress signals carried a lethal penalty. A screaming hominid infant or a wailing injured adult served as a loud acoustical beacon, instantly attracting the attention of nearby deadly threats. As humans developed longer childhoods and more complex social structures, the silent signal hypothesis suggests we evolved to decouple distress from sound. We needed a way to signal for help that didn't alert every predator in the area. Visual tears allowed individuals to broadcast extreme vulnerability directly to proximal allies and caregivers. It became a targeted method of transmission, ensuring that only those close enough to help could receive the signal. Tears provided an acoustic compromise, a localized distress beacon that ensured the survival of early tribes by prioritizing safety over volume. Beyond the visual signal, tears serve an even more subtle biological function. Popular culture often champions the catharsis theory, the idea that crying physically excretes toxic stress from the body, like a pressure valve. Real-time psychophysiological studies show the opposite. The act of crying actually induces immediate physiological stress and spikes the heart rate. Any sense of relief is usually delayed by 30 minutes and is almost entirely dependent on receiving support from the people around us. Recent research explains why this support is consistent. Human tears contain perceptually odorless chemosignals, functioning as an advanced invisible signaling system. When inhaled, these signals bind to olfactory receptors, triggering a brain response that significantly reduces reactive aggression. Tears act as an externalized extension of our nervous system, providing an invisible olfactory signal that dampens hostility in those nearby and guarantees the authenticity of our vulnerability. Weeping is the ultimate social glue, an involuntary biological language that compels the human group to connect, protect, and survive together.